Hello dear students, hope you all are doing well. Today we will begin with chapter number 8, How do organisms reproduce? But before coming to this chapter, I would like to ask you one question. What makes our planet Earth so different from other planets? Well, I know your answer would be only on Earth there is life, isn't it? Now my second question is, name the process which is responsible for the continuity of life on Earth. And I know your answer would be the process is reproduction. So in this chapter, we will be dealing with this process of reproduction. So as I told you, reproduction is the process which is responsible for continuity of life on Earth. But in this chapter, we will be dealing about the process of reproduction. So the name of the chapter is How do organisms reproduce? In this chapter, today I will be dealing with all these topics. We will be studying about the definition of reproduction, and we will be correlating that with DNA replication. Then we will be studying about the significance of DNA replication along with reproduction. After that, we will be studying about the modes of reproduction, and then finally we will study about the asexual modes of reproduction in detail. So let's begin with the first topic, reproduction. So what is the definition of reproduction? Reproduction is a process by which the organisms produce new organisms of its own type. So to understand this, we must know that all living organisms have a definite body design, and the most basic body design is that of a cell. Suppose this is an organism having a single cell, that is unicellular organism. And you know, all the organisms have their body design, the information inside the nucleus, because nucleus is the brain of the cell, you know it. And inside the nucleus there are chromosomes. But these chromosomes are made up of a wonderful molecule which is DNA, deoxyribonucleic acid. So all the information about the body design is present in this DNA. These DNA molecules are responsible for the production of proteins, and because of these specific types of proteins, there is a specific body design. So reproduction is actually nothing but the replication of this DNA along with the replication of the body design. So you can see this is one mother cell. This will undergo replication first in DNA. DNA will replicate, and then it will also replicate its cellular apparatus. So we can say that reproduction is a process of DNA replication along with the replication of the cellular apparatus, so that the new organisms which are produced, they have similar DNA as their parent, also similar body design. Okay, now the question arises, if there is DNA replication, why all organisms are not alike? Why there are so many differences in the organisms? Well, the answer is in the process of DNA replication. Though DNA replication tries to replicate a similar DNA as the DNA of its parent, but these processes might occur certain errors. So there may be certain errors in DNA copy which may be resulting into the variations in the new organisms. Are these variations good or bad? Well, the answer is both. Most of the times, these variations may be very dangerous for the organism and the organisms may die. But few variations, they might be of some advantage to the organism. For example, I will give you one condition. Suppose there is a water body in temperate area where the temperature is low, and in that water body there are many bacteria present. So these bacteria are adapted for lower temperature. If there is global warming and because of the global warming the temperature of the water has risen, so what will happen? In that case, most of the bacteria will start dying because they were not adapted for the higher temperature. But sometimes due to mutations, certain bacteria have developed an adaptation to survive in hot water. So these bacteria which have developed these variations are because of error in DNA copying, error in DNA replication. So this error which has produced some variations is advantageous to these bacteria, and only those bacteria which are tolerant for higher temperature, they will survive and other bacteria will die. So in this way, variations, if they are very subtle and mild and they are not harming much to the organism, these are helping the organisms to evolve and they become the basis for evolution. Okay, so I hope you have understood the relation between this reproduction and the process of DNA replication. Now let's move to the next topic, that is modes of reproduction. What are the different methods by which organisms they produce new organisms of their own kind? So we can categorize all of these reproduction methods into two main categories. First is asexual mode of reproduction and second is sexual mode of reproduction. So let us find out the difference between them. Asexual mode of reproduction is called as monoparental, which means it involves only a single parent. Whereas the sexual mode of reproduction is biparental, where two parents are involved. And here, because there is a single parent, all the new organisms which are produced, they are just similar to their parent and they are called as clones of their parent. Whereas in case of sexual reproduction, because two individuals are there, so the new organisms which are produced, they are recombinants of their parents. So they show variations. Okay. Then in the process of asexual mode of reproduction, it is very simple process. So there is no gamete formation, no fertilization, no zygote formation. Whereas here, it's a complex process which involves gamete formation followed by fertilization and which results into zygote formation. And this zygote undergoes embryonic development and a new organism is produced. So this process is quite slow and very complex, whereas asexual reproduction is a very fast process and it is very simple as well. So these are the differences between asexual mode of reproduction and sexual mode of reproduction. Now let us see the various types of asexual modes of reproduction. I have noted down all the different types of asexual modes of reproduction, which are binary fission, multiple fission, budding, fragmentation, regeneration, spore formation, vegetative propagation, and micropropagation. We will be studying them one by one. So let's begin with the first type of asexual mode of reproduction, which is binary fission. Now let's study all these asexual modes of reproduction one by one. We will begin with fission. I have mentioned two types of fissions. One is binary fission, other is multiple fission. The meaning of the term fission means breaking. When a single organism, because this fission takes place only in unicellular organisms, when the organism which has become mature and it breaks into two, that is called as binary fission. But when the organism breaks into many parts, that is called as multiple fission. So, in case of binary fission, which is very common in amoeba and also in leishmania. What is the name of the disease caused by leishmania? It is Kala Azar, that is black fever. So, both amoeba and leishmania, they show binary fission. But there is a slight difference. Like in case of amoeba, because amoeba is irregularly shaped. So, the binary fission is transverse type. Whereas in case of leishmania, the binary fission is longitudinal type. Like it will divide only from a specific plane so that it longitudinally splits into two parts and then these two parts develop into new leishmania. So this is binary fission. Next is multiple fission, which is very common in Plasmodium. What is the name of the disease caused by Plasmodium? It is malaria. So the malarial parasite multiplies by multiple fission. Generally in our body, in human body, it divides asexually into two parts, into two body organs. One is our liver. It enters our body, goes into liver and divides by multiple fission. After that, it comes into the blood, enters into our RBCs and again divide by multiple fission. So, this Plasmodium, first it develops a thick wall around it, which is called as cyst. And then inside the cyst, the cytoplasm divides into many parts. And when it has divided into many parts, this cyst breaks open. And all these small daughter cells, they are liberated, which grow into the new Plasmodium. Okay, so this is multiple fission. Both binary fission and multiple fission occurs in only unicellular organisms. Now, third process is fragmentation, which occurs in multicellular but very simple organisms, like in case of Spirogyra, which are very commonly found in ponds. This Spirogyra is ribbon-shaped, filamentous structure. When it grows very long, it starts breaking into small fragments. And these fragments later on retain their original size. And this process is called as fragmentation. Okay, so these are the three processes. Next process is budding. So this budding is very commonly seen in yeast and hydra. These organisms, they start growing some outgrowths on their body, and when these outgrowths detach from them, they develop into new organisms. Same way in case of hydra also. When the outgrowth grows in size and detaches from the parent body, it develops into a new organism. Now this budding is quite different from fission. The main difference is that in both multiple fission, binary fission as well as fragmentation, parental identity is lost, which means there is no parent after the process, because the parent only divides into two or more parts and develops into new organisms. Whereas in case of budding onwards, parental identity is maintained. So parent can keep on producing new outgrowths and which develops into new organisms. Okay, so this is about budding. Next is regeneration. Though we are taking regeneration as a mode of reproduction, actually it's not a mode of reproduction. Regeneration is just a restorative process when, because of some accidental reason, some organism has lost some part. So those organisms can regain those lost part. But few organisms like planaria, starfish, their body is so simple that the broken parts can develop the complete organism. For example, if you see this planaria, suppose accidentally this planaria is divided into three parts. It is cut off into three parts. So all these three parts can regenerate the lost part. So from one planaria, after cutting into three pieces, three planaria can develop. So this is called regeneration. Regeneration is quite different from fragmentation. Fragmentation is a normal method of asexual reproduction, whereas regeneration is not a method of reproduction. Fragmentation is done by very simple organisms, whereas regeneration is generally done by complex organisms. There are specific cells which have the ability to regenerate. Okay? Now next is spore formation. Spore formation is a characteristic feature of fungi, which is very commonly seen in rhizopus, bread mold. You must have seen, especially in these seasons like rainy season, there is dampness, lot of moisture and lot of heat. So because of this hot and humid weather, sometimes you might see some kind of fungus growing on the bread. So that is called bread mold. So these bread mold, they grow asexually by the process of spore formation. These spores are dispersed in the air. They have thick wall which protects them from unfavorable condition, and whenever they find favorable condition, they settle and start growing the hyphae. These hyphae, thread-like structures, will develop some outgrowths, and inside these outgrowths there are these spores. These spores are produced in large number and they keep on remain suspended in the air and get whenever they get the favorable condition and they grow. So this is spore formation. Now next method of asexual reproduction. Now next type of asexual reproduction is vegetative propagation, which is shown by plants. Plants reproduce both by asexual method, that is vegetative propagation, as well as sexually with the help of seed. But there are certain advantages of vegetative propagation over the sexual reproduction. What are they? The first advantage is that vegetative propagation is a very fast method. So the plant does not have to wait for the seed formation for developing into new plant. Any vegetative part can be broken off from the main body part of the plant and it can grow into a new plant in very short period of time. Second, even the plants which do not produce seeds, they can propagate. For example, you very rarely might have seen rose plant, but it propagates so fast. It is mainly by cutting. Like these are the plants, these are the stem cuttings of bougainvillea. Have you ever seen the seeds of bougainvillea? No. They are generally growing by vegetative propagation through stem cutting. Likewise, there are many parts of the plant which propagates through vegetative propagation. For example, roots. Plants like dahlia, their roots can be used for propagating. We don't require their seeds. Likewise, stem. I showed you the stem. Also, I will show you two more stems. This is one stem. It's not root, it's ginger. And this is potato. You can see these potato have got small outgrowths. These are the buds, these are called eyes. From eyes, the buds will grow and they can grow by vegetative propagation into new plant. If I cut it into 5 or 6 pieces, those 5 and 6 pieces will develop into new plant. Likewise in case of ginger. Similarly, bryophyllum. I will show you a very beautiful plant. It's a leaf, bryophyllum. Can you see? How beautiful. They have got small plants attached to them. You can even see their roots. So these plants, they can just detach from these and they will grow into new plant. You can see this. So this, when it will fall on the ground, it has already developed the roots. It will develop into a new plant. This is bryophyllum. You can see this. Okay. So vegetative propagation is a very fast method of asexual reproduction in plants. Next is, the last method is micropropagation or tissue culture, which is done in labs. There are specific tissue culture labs. Some plants, basically very rare ornamental plants, medicinal plants, as well as genetically modified plants, which don't even grow by vegetative propagation. They can be grown in the lab in specific culture media where any tissue can be taken and grown in that culture media, and that tissue will develop into a mass called as callus. This callus will now, if it is grown further, it will develop into a cluster of small plants, and you can separate those clusters and can, from a single small piece of tissue, you can grow millions of plants. So that is called micropropagation or tissue culture. So with this, we have completed the complete asexual reproduction in organisms. Let us now recapitulate and summarize the whole process once again quickly with the help of a small animation. Now we know that reproduction is a process which involves DNA replication along with the replication of the body design. So this is a molecule of DNA. This molecule will undergo replication and then it will also replicate its body design. Okay.
Now this reproduction is of two types, you already know, asexual and sexual. In asexual only one parent is involved, whereas in sexual reproduction two parents are involved, and the organisms which are produced after sexual reproduction, they show variations, whereas in asexual reproduction there is no variations. But today we have already studied different types of asexual reproduction. We will just quickly recapitulate them. First one is binary fission, which is shown by unicellular organisms like amoeba. Amoeba, as you know, they are irregularly shaped, so they can show binary fission from any plane. So this type of binary fission is called as transverse binary fission, whereas organisms like Leishmania and Paramecium, they have a specific body design. So they also show binary fission, but only from a specific plane, that is called longitudinal binary fission. Same way multiple fission is shown by Plasmodium, where a single organism can produce a large number of clones. Okay. After this, the next type of asexual reproduction which you have studied is fragmentation, where in case of Spirogera, a small fragment which is produced can develop into the complete organism. So in this way, organisms reproduce very fast. Next is the regeneration process. Though regeneration process, as I told you, it's not a reproduction process. It's only a restorative process. In this process, the part which is cut have got specific cells which can restore the lost part. Next one is budding. As in case of Hydra, Hydra can produce an outgrowth. This outgrowth later on can develop into a new organism. Next is vegetative propagation. There are many plants which do not produce seeds. They propagate through the vegetative propagation. This is grafting. Grafting is also a method of vegetative propagation. I showed you the bryophyllum. Look here. So in this bryophyllum, there are small plantlets which are present on the notches of the leaves. When they fall down, they germinate into new plants. Likewise, there are many other plants which have got parts which grow for vegetative propagation. Like you can see, there are so many: potato, ginger, onion. So these parts, they have the ability to grow by vegetative propagation. Same in the case of this potato. If they have small outgrowths, buds, if you cut them into 6 or 7 pieces, each piece is able to grow into a new plant. After vegetative propagation, see, vegetative propagation has got many advantages. What are they? Let's recapitulate again. It is a very fast method, much faster than sexual reproduction, and it requires only one plant. There are many plants which have lost the ability to produce seeds, so these plants can propagate through vegetative propagation. Also, if we have a very good variety of any plant, we can make its own clones, like these clones will be genetically similar to the parent plant. So by the vegetative propagation, we can make the clones of very good ornamental plants, and these clones will be genetically similar to the parent plant. Okay, now let's move to the next type of asexual mode of reproduction, which is very commonly seen during rainy seasons, as in the case of bread mold Rhizopus. It grows on the bread with the help of thread-like structures called hyphae, and these hyphae grow outgrowths which develop into sporangia, and these sporangia produce very thick-walled spores. These spores, when they find a suitable medium, they again start germinating by growing new hyphae and spores. Now, the last type of asexual mode of reproduction is a highly advanced and scientific, that is tissue culture. In tissue culture, we just require a small piece of tissue which can be grown in the artificial medium, in culture media. This part will divide mitotically to produce a lump of cells, undifferentiated mass of cells called callus. This callus will differentiate and produce a large number of plantlets. So this is a very fast method of growing new plants. So after doing all these things, I am sure you are able to answer questions. So these are your assignments. These questions are very simple. You have to answer. First is list the modes of reproduction in living organisms. Question 2, explain how the mode of reproduction of an organism is related to its body design. Next question, explain how unicellular organisms reproduce by fission. And last question, illustrate the types of asexual reproduction in multicellular organisms. So you have to explain all: fragmentation, regeneration, budding, vegetative propagation, and spore formation in your notebook. I hope this topic is very clear to you. Thank you.